Hello everyone, welcome to our video about the Lac Operon. As you can see in the picture, there are several parts to the Lac Operon. There's the cap site, the promoter, and the operator, and as well as the genes that are part of the Lac Operon. In this video, we will talk about what each part does.
Let's first start out by talking about the promoter region. The promoter region can be bound by an RNA polymerase, which has the ability to transcribe genes. The product will be an mRNA strand, which can then be translated by a ribosome to make protein. In the case for the Lac Operon, the protein transcribed are associated with the digestion of lactose.
Next, we have the repressor. The job of the repressor is stop transcription from happening, as you can see by the X on the little red arrow. What we have here in the green is allolactose. So when there's lactose in the environment, there's also allolactose, and allolactose is an inducer of repressor, which means that it will bind to the repressor and remove it from the operator site, which allows the transcription to go on, as you can see by the red arrow.
Next, we have the cap site. This site is where the cap proteins are able to bind. The cap protein is activated with cyclic AMP, shown in yellow. It binds to the cap protein and activates cap, which then could bind to the cap site. Once bound to the cap site, the cap protein is able to bind to the RNA polymerase, which will then allow for efficient transcription to happen.
Now we're going to examine the Lac Operon under 3 different conditions. The first condition is glucose only, the second condition is lactose only, and the third condition is lactose plus glucose in the environment. So first, let's look at how the Lac Operon works under a glucose condition. This is simple because when glucose is in the environment, the Lac Operon does not get transcribed at all. When there's high amount of glucose, there's low amount of CAMP, which means that the cap protein will not be activated. The repressor stays on the operator because there's no allolactose in the environment to remove it. So this is how the Lac Operon works under the glucose condition.
In the condition where only lactose is present, allolactose will be floating around. Keep in mind that allolactose is an isomer of lactose. As mentioned before, the allolactose, which is the inducer, will bind to the repressor that's bound to the operator, and this complex will leave the operator. And as a result, RNA polymerase will not be blocked and will continue to transcribe. Over at the cap site, we see that there's a cap protein that is activated and bound to it. This is because when there's no glucose around, there will be a high amount of cyclic AMP, which is shown in yellow. This cyclic AMP would then bind to cap and activate it, making RNA polymerase much more efficient when transcribing the ZYA genes. These genes would later be encoded for proteins that are associated with the degradation of lactose.
So now let's examine how the Lac Operon works in a condition where there's both lactose and glucose. Since there's lactose in the environment, there will be a derivative of lactose in the form of allolactose, which binds to a repressor and removes it from the operator site, as indicated by the black arrow. This enables the RNA polymerase to transcribe the Lac Operon. Since there's high amount of glucose in the environment, there will be a low amount of CAMP, which means that the cap protein will not be activated. So the result of an environment is that the Lac Operon will be transcribed, but there will be very, very low levels of it, since the preferred source of energy for the body is glucose.